Hi, I'm J Phelan. Let's learn some biology. Recall when we were talking about Hardy-Weinberg equilibrium and Hardy-Weinberg equations that these were initially thought up in 1908 because biologists were perplexed by the question of whether or not dominant traits should gradually increase in frequency in a population, or why recessive traits should gradually decrease in frequency, should disappear from a population. Somehow, and I think it's just because of the words that were used, people imagined that was the case. And a mathematician in England, Hardy, and a biologist in Germany, Weinberg, independently came up with this idea where they discovered that, in fact, no, dominance or recessiveness doesn't influence whether a trait increases its frequency or decreases its frequency in a population. So that was the origin of what we'd call the Hardy-Weinberg principles. But what was more important about them is that they gave us a way of viewing a population from a genetic perspective. In other words, thinking about genes not as a set of instructions that code for a protein. No, now it's about over time. Are the allele frequencies in a population increasing or decreasing? So we're not really thinking about individuals. Two important insights that come from the Hardy-Weinberg principles include, one, if we know the genotypes of the individuals in a population, we can figure out what the allele frequencies are for that population. Why would we want to do that? Well, because evolution is a change in allele frequencies in a population. So you have to have a time 1 and a time 2 to know if they've changed. So how do we figure them out? We just add them up. Second insight that came from the Hardy-Weinberg principles is that if we know the allele frequencies in a population, then we can make predictions, we can compute what we would expect the genotype frequencies to be in the next generation. And an important qualification on those predictions are that that only holds true if there's random mating in the population and if no evolution is occurring. When those are true, those are called the Hardy-Weinberg assumptions, then from those allele frequencies, we can make the prediction about the genotype frequencies. I want to look today at a couple of examples where we use these principles to look at how evolution can work in a population, to look at how random mating can influence the genotype frequencies we see, and so on. Let's take a look at some what we call population genetics data. We have genotype frequencies from two different populations. Population 1, we see 0.36, 0.48, 0.16. These are the frequencies, the percentages, if you will, of the three different genotypes if we're looking at a single gene that has two alleles. Now remember, you don't necessarily have a single gene for a trait with two alleles. You could have 3 alleles, like the blood groups do, or 4 alleles, or 5 alleles, or a whole bunch of alleles. For our purposes here, we're going to look just at one gene, 2 alleles. Population 2 has a different set of genotype frequencies, 0.55, 0.10, 0.35. We can ask this question, what are the allele frequencies in these two populations? And we can also ask, are the populations in Hardy-Weinberg equilibrium? And we'll see why that matters or what it means to be in Hardy-Weinberg equilibrium or not. For the first population, how do we do it? Remember, whenever you want to know the allele frequencies from the genotype frequencies, you just add them up. So if 0.36 is the frequency, so the percentage of big A, every one of those individuals has 2 copies of the A. So we'll say 2 times that number. And of the heterozygotes, there's 0.48 is the frequency. Every one of those individuals has 1 copy of the big A. So we take 2 times the 0.36 plus 1 times the 0.48, and we divide it by the total. Well, here, because the 0.36, 0.48, 0.16 total up to 1, it's a frequency. Remember, every individual has 2 alleles, so we divide that by 2 to figure out the frequency of each. So the math works out, 1.2 over 2. The frequency of the big A is 0.6. You can think of that as its market share. And if its market share is 0.6, and there are only 2 alleles in the population, they have to total 100% or 1. So the other is just 1 minus the 0.6 or 0.4. If you wanted to figure out what the frequency of little a is, you do the same thing. 2 times the frequency of little a plus 1 times the frequency of the heterozygote divided by 2, you get the same thing. So those are frequencies of the alleles in population 1. Now let's look at population 2. Population 2, we do the same thing. big A occurs 0.55 or 55% of the time. So 2 times 0.55 plus 1 allele in each of the heterozygotes, 0.1, divided again by 2, we get 0.6. If that's 0.6, the frequency of big A, then the frequency of little a has to be 0.4. They're the same. Same frequencies of alleles in the population. If you view the population as just a cauldron of alleles, there's no difference. But in the two populations, they've distributed themselves into individuals, into different genotypes, differently. That can happen, and let's look at that. So we want to understand, is the population in Hardy-Weinberg equilibrium? Well, we know the allele frequencies, so now we can take those and say, what are the expected genotype frequencies? If you have a 0.6 and 0.4, how should they distribute themselves? If there's no evolution, there's random mating. Well, we know how to do that. Frequency of big A is just the probability of getting a big A from one parent, that's 0.6. The probability of getting a big A from the other parent, its frequency is 0.6. Multiply those together. It's p squared, 0.6 squared, 0.36. The probability of getting a big A and a little a, in other words, what we expect the frequency of the heterozygote to be, is just going to be 2 times p times q. So you have to get the big A from one parent and the little a from the other parent, or vice versa, the little a from that first parent and the big A from the other. Two ways to do it. When we multiply it out, we get 0.48. And same thing for the expected frequency of the genotype little a. It's just the probability of getting it, 0.4 times 0.4. You have to get 2 of them, so 0.16. With allele frequencies of 0.6 and 0.4, we expect to see genotypes in this frequency, 0.36, 0.48, 0.16. Are the populations that we looked at previously in Hardy-Weinberg equilibrium? They are if what we expected is what we observe. So we have to compare these expected genotype frequencies with what we observed in nature, in these populations. So we looked, here are the observed frequencies. Then we look at our expected ones, 0.36, 0.48, 0.16. It's clear to see that population 1, yes, the alleles have distributed themselves into genotypes exactly as we would predict. So yes, that population's in Hardy-Weinberg equilibrium. Population 2, even though it has the same allele frequencies, we don't say that it's in Hardy-Weinberg equilibrium because what we observed is different than what we expected. And there's a question that's relevant to this. What happens if you don't have random mating? Random mating imagines the likelihood of a gamete that has a big A fertilizing an egg that has a big A or a little a, that they are at random. The only thing that's going to determine how often it happens is the frequency of each in the population. But what if individuals are looking and thinking, I'd rather mate with you. I'd rather mate with you. Something like that. Or I'd rather not mate with you. All of a sudden, non-random mating, we can come up with some different genotype frequencies for a given set of allele frequencies. So looking back at our two populations, look, in population 2, we got 0.1 as the frequency of the heterozygotes. We don't see very many of them, given the 0.6 and the 0.4. Why is that? Well, sometimes if whatever the phenotype is of big A, or little a, they would rather mate with an individual that has a like phenotype, then we're going to see a clustering. We're going to see more of the big A, more of the little a, and that doesn't leave as many to come together in the heterozygote. Or vice versa. Individuals may prefer to mate with someone as different from them as possible. And if that's the case, then we'd see way more heterozygotes and fewer of the homozygotes. And we can do that. We can redistribute them while still having the 0.6 and 0.4. But that's not random mating. So, important take-home message is that if you observe genotype frequencies and they're not what you expected based on the allele frequencies that you've calculated, the population's not in Hardy-Weinberg equilibrium. That doesn't mean it's a useless concept. It doesn't mean that you've done something wrong. It just means that one of our assumptions was violated. There's either been non-random mating or maybe evolution's occurring. Both of those can lead to a difference between the expected and observed. So I was suggesting that maybe non-random mating was going on here, but what you do is you look at your populations now. It gives you guidance towards new research. that you'd want to do. You'd want to say, let's take a look at who's mating with whom. Is it random or is it not? Or maybe the heterozygotes were dying at a higher frequency. That would be evolution. That would be selection. Let's look at an example where that's the case. We don't know based on this earlier example. All we know is that one of the assumptions of Hardy-Weinberg equilibrium was violated. So we'll add some more realism now to Hardy-Weinberg principles. If we have, again, we're using the simplistic single gene with 2 alleles, even though all these equations have been worked out for 3 alleles or 4 alleles. Instead of a P and a Q, you've got a P and a Q and an R, or a P, a Q, an R, and an S. The computations get cumbersome, but they're not that hard. Still 7th grade math. If we have big A and little a, our expected frequencies of the genotypes are going to be P squared, 2PQ, Q squared. But let's add in some realism. Every genotype has a fitness. And remember that fitness is your relative reproductive success compared to individuals with the other phenotypes or genotypes in a population. Your relative reproductive success compared to them in a particular environment. In other words, in some environments, certain genotypes aren't going to do as well. So let's look at an example here. So I've taken each of these, P squared, and I've multiplied it by W, which is its fitness for big A, or 2PQ, I've multiplied it by W for big A, little a, the fitness of that one. And people use different ways of giving a number to fitness, but it's a relative measure of your reproductive success. So a common way to do this is to have the most fit genotype be 1. And the others are going to be less than that. They're not reproducing at as high a rate as that other one. So we can add in the fitness. I'm imagining for this example, incomplete dominance. In other words, you can see a different phenotype for the big A, the heterozygote, and the little a. But I'm also imagining that the big A has the highest fitness. The little a, most of them are dying. We're calling the fitness 0.2. So 80% of them are going to die. And the intermediate, the heterozygote, we're saying that the fitness for them is 0.8. In other words, having a copy of little a is not so good. You're not going to reproduce at as high a rate as if you didn't have a copy of little a. So we give these fitnesses to the genotypes, and it might be because we're looking at directional selection by a farmer or an animal breed or something like that. I'm going to propose for this population some genotype frequencies. We'll say that the frequency in the population is 0.64 for the homozygous dominant. We'll say it's 0.32 for the heterozygote and 0.04 for the homozygous recessive. We could propose anything, but I'm giving those from those genotype frequencies. Remember, our first insight from Hardy-Weinberg is we can always calculate what the allele frequencies are. We just count them. So we take 2 times the first one plus 1 times the second, divided by 2. We come up with 0.8 and 0.2. 80% of the alleles are big A, 20% are little a. That's before we've done any of the selection. Those are just what the allele frequencies are. Selection, remember, eliminates individuals. So it eliminates genotypes from the population. In order to incorporate this into our view of the world, we take those genotype frequencies and now we kill off some of the individuals. So for the 0.64 that we saw of big A, that fitness was 1, so we just leave that as it is. That's still 0.64. But the heterozygotes, only 80% of them make it. So you take the 0.32, multiply it by the fitness, 0.8, you get a new number, 0.256. And for the homozygous recessive, remember, most of those die off. They're being selected against. 0.2 is their fitness. So we take the 0.04 times 0.2, and we get a new number, 0.008. So we've removed some individuals. But we still want to know what the genotype frequencies are. And here's that step that can confuse people. It's a trivial step. It's just mathematical bookkeeping. That 0.64, 0.256, 0.008, those aren't true frequencies anymore. Because remember, a frequency is when you have some number as a proportion of the whole. Well, we've removed a whole bunch of individuals, so we have to recalibrate it. So we take, if we add up the 0.64, 256, 0.008, we get 0.904. In other words, we've gotten rid of about 10% of the population. So now we divide each of these numbers by the whole to get the actual frequency of that genotype. Turns out to be a little higher. We've gotten rid of, so the big A is really, it's 0.708. How did its frequency rise? Well, you get rid of some of the other ones, its market share increases, right? They total 100%. For the heterozygote, it turns out to be 0.283, and for the homozygous recessive, it's 0.009. In other words, it used to be 0.04, now its frequency is 0.009 because we've gotten rid of 80% of them. We want to know the new allele frequencies. We have genotype frequencies, and remember, first insight from Hardy-Weinberg, we can determine them. How do we do it? Count the alleles. So we take 2 times the frequency of the big A, and 1 times the frequency of the heterozygote, divide it by 2, and we're going to get some number. So that turns out to be 0.85. Frequency of little a is just going to be 1 minus that, 0.15, or you could do it the hard way. You could say 2 times the frequency of the homozygous recessive plus 1 times frequency of the heterozygote. Still works out the same. So our new allele frequencies, 0.85, 0.15. And we don't want to get lost in all the math and the numbers. These are grounded in reality. With incomplete dominance and directional selection, initially we had 0.8, 0.2. And then after selection occurred, what happened? Frequency of big A, 0.85. Frequency of little a, 0.15. They've changed. If you have variation in fitness, in other words, some of the genotypes aren't as good as others, and you're removing them, you're doing selection. The allele frequencies are changing. And remember, what's the definition of evolution? A change in allele frequencies in a population. And in this case, we've done directional selection. We've removed some, so of course they change. But it's kind of nice to see here that the 0.8 goes up to 0.85 because the little a was not doing so well. So it benefits. The little a goes down. Over time, natural selection in a population, or it might be artificial selection that a farmer's doing, an animal breed or something like that, when you are selecting against certain genotypes and removing alleles, you reduce their frequency in the population. You can even cause them to go to 0. You can lose alleles. You can end up with fixation. At that point, you're not going to make any further progress in doing your selection. It's an absorbing boundary, they say. But you have to keep in mind that that's it. If we define evolution as a change in allele frequencies, we do some evolution in a population. Of course, the Hardy-Weinberg equations and principles are going to show us that the allele frequencies have changed. So it's maybe a little counterintuitive, right? Because Hardy-Weinberg has some assumptions, random mating, no evolution. But as we've seen here, we can still use those equations for viewing the population, for viewing changes in the population, if you have non-random mating, or if you have selection occurring.